Let's say all of that is 100% genetic. It's not, it's just an example, but as an example, let's pretend it's 100% genetic and both parents have the gene to be good at music. Well, then they probably live in a household that has more instruments, that plays more music, they consume more music, things like this. So these children who have that genetic trait are growing up passively in an environment that supports their genetic trait. So when you go and measure and say how much of this is the gene versus the environment, how much is the nature versus nurture, it's kind of hard to tell, right? Because they're in an environment that passively supports their genetics. And that's why it's called a passive genotype environment correlation. So that is when the organism, or the children in this case, are not making any kind of change or impact to their environment. It's just already in a way that passively supports them. That's a little bit different than the evocative genotype environment correlation. This is when an individual's characteristics make a change in the environment unintentionally. So let's pretend that friendliness was 100% genetic. You take a kid who's got 100% of those genes and is very friendly, and then another one who has none of the genes and is very unfriendly. Well, the one that's very friendly will probably end up getting more friends. They will change their environment because they are friendly. More kids will want to be their friend. They'll get more practice with social situations versus the one who's got none of the genes for friendliness, probably has fewer friends, fewer chances to practice social scenarios. And so with that, they have changed their environment because of their behavior unintentionally that they have elicited. They are evoking a certain environment to them. And that's why it's called the evocative or evoking or eliciting, the evocative genotype environment correlation. Again, kind of like the passive one, just makes it harder to tell, is this genes or is this nurture, right? Is this genes or environment? The last one is active genotype environment correlation. It's similar but as the evocative, but in this one they are picking a situation. So let's pretend parkour ability, dance ability, like kinesthetic ability with your body is 100% genetic. If that was the case, you might choose to go and play at a place like this instead of somewhere less active, right? So if that is 100% genetic, again, it's not, it's just an example, you might actively pick a niche. The things that you pick might be supporting your genetic traits. So that's active genotype environment correlations, or picking a niche. So those are some of the ways both passive genotype environment correlations, evocative and active genotype environment correlations can make it a little difficult to piece out how much of this is genetic versus environment because they are working together for all three of these examples. So another thing to add on to this is epigenetics. So epigenetics is the interaction of your genome based on the environment. So with this, there's a couple examples in a video that I want to show you. So rather than this slide, I'll make the main points. That epigenetics is about changing the way those proteins read the structure of the DNA because of environmental inputs. So this slide is worded poorly. I should have fixed that. So it's because it's not changing the DNA sequence, it's not changing the code, but it is changing how that code is read and used by proteins. So again, this is another example of bidirectional interaction. Our environment can affect the way our genes are read, the way our genes are read can affect how we interact with our environment, and vice versa, and it's a continuing spiral. So let's look at some examples from a textbook. This gene, the 5-HTT-LPR, only activates and causes depression when you have a stressful life. So if you have this gene and you have a not stressful life, you are less likely to get depression. If you have this gene and you do have a stressful life, you are more likely to get depression. If you don't have the gene at all, well, then you're probably not going to get it or you're less likely. Not probably not. You're less likely. There's other things that can cause depression. It's not just this single gene. Again, genes in general collaborate together. So let's watch a couple videos with a couple really interesting examples about epigenetics to hopefully make this relationship between your environment and genes a lot more clear. So let's hope this SciShow one does not copyright strike my this video. That has happened before when I've used this as an example, as well as this example. So we'll use quite a bit of this one and then a couple examples from this one.
Say that a clone or a secret identical twin, because you know that a twin or a clone is a genetically identical person to you, which is crazy. But it's true. Anyway, so you have this clone or your twin or what have you, and your parents decide that it's too much trouble to keep both of you. And so they give your clone to a kind of sketchy traveling circus. Now, I know your parents probably wouldn't actually do this, but just roll with me for a minute. So, for purposes of this example, let's just say that you grew up living a normal suburban American life, while your clone grew up with less stability and less access to nutritious food and less education, but more exercise and more access to sword swallowers and bearded ladies. And let's say you guys are 50 years old or whatever, you get together and have some lunch. So you're sitting there across the table from your clone. What do you think you see? Well, for starters, Clone probably has a tattoo on its face. And if Clone had stayed in the circus, Clone probably, professionally rides a unicycle over a tightrope, so Clone is probably in pretty good shape. But then again, Clone probably smokes cigarettes and probably was malnourished as a child, so might be slightly shorter than you. By the same token, let's say that you've been eating Sonic tater tots 5 days a week since 1992, so you're looking a little bit tubby. Plus, you spent the last 20 years at a really stressful job that puts you at risk for all kinds of weird health complications. So at 50, you and Clone would probably look pretty different from one another. But it turns out you don't just look different, you actually have become different. Now, some scientists looked at your genetic code. They would say that your DNA was still exactly the same as that of the person. That is to say, if your genome is a paragraph, all of the letters would be in the exact same order. But
all right, so before they continue with this analogy, or is it a metaphor? Either way, before they continue on with that, let's look back at the DNA strand image that we have here. So here is the DNA strand. So what they're saying is that this genetic code is all still in the same place. You and twin both have the same exact order of this DNA molecule. So all of your cells, you measure them, they are identical still. So the order of the ATCG is all still the same. So they have G, this is kind of hard to read, but GTG, then here GTG, CAGACT, right? So their order is still the same as your order. The actual molecule of DNA doesn't change the order. So the changing of that order, that would be a mutation. That's not what epigenetics is doing. The DNA strand stays the same. It's this step, the protein reading or doing something based on the code, that is changing. And so let's see some examples of how that happens in the body. I'll rewind to the example from the paragraph. So you and twin are pretty identical. Your genes are identical. And let's go back to this example.
If your genome is a paragraph, all of the letters would be in the exact same order. But on another level, there would be a huge difference. And if we extend this metaphor, we can say that the letters are in the same order, but the spaces and the punctuation are all in different places. Of course, completely potentially changing the message of that paragraph.
So that is what we're talking about, is the reading of this paragraph is going to be different. The reading of this gene is different because of epigenetics. So epigenetics makes the protein do something differently than you might otherwise expect without that epigenetic factor to do something in the body. So we'll learn about two major processes. Here we'll learn about methyl groups and histone patterns. And if you want to watch the whole video, it's got some other really good examples, talks a little bit about the history of this research, which is great, but we'll actually see a couple more concrete examples from the other video. But let's learn about methyl groups and histone patterns.
The study of this genetic punctuation that I'm talking about is called epigenetics, which literally means above genetics. The epigenome doesn't change your DNA, but it decides how much or whether some genes are expressed in different cells in your body. Epigenetics looks at what happens to your genes over the course of your life and whether those changes could be passed down to your children or even your grandchildren. So here's the way epigenetics works. You have billions of cells in your body, and they each contain your DNA, the same exact blueprint of your genetic code. But just because they have the DNA doesn't mean that they know what to do with it. They need outside instruction from these little carbon and hydrogen compounds called methyl groups. The way these methyl groups control the genome is by binding to a gene and saying, do not express this gene. The methyl groups bind differently to your genome in a skin cell versus, say, a tongue cell or an eyeball cell. And that is one of the ways that a cell knows that, hey, I'm a skin cell, or hey, I'm an eyeball cell, or hey, I'm a muscle cell. In addition to methyl groups, epigenetic.
All right, so with those methyl groups, you could think of it, it is actually a physical process on the DNA that these are other molecules binding to the DNA. And so essentially you could think of it as it's blocking some of the DNA from being read by proteins. So you could say like it's a big X up here, like don't read these genes. That's not important. You are becoming a brain cell. You are becoming a skin cell. So there might be a big X up here, a big X down here. And so the protein comes and globs onto the DNA and says, what are my instructions? When it hits the place without instructions, well, it can find the correct instructions because the instructions for brain cell are not the same as the instructions for skin cell. So when it comes to get its instructions, it will know what to do. The protein will read it differently in the different cells.
X is also controlled by histones, which are proteins that are basically the spools that DNA winds itself around. Histones can change how tightly or loosely the DNA is wound around them. If they're more loosely wound, the genes can express more, and if they're more tightly wound, then they express less. So whereas the methyl groups are more like a switch, the histones are more like a knob. Every cell in your body has a distinct methylation and histone pattern. And that is what gives every cell its marching orders. Think of your genome, the DNA, as the actual hardware of the computer, while the epigenome is more like the software, which tells the hardware what to do. The genome is what's going to be doing all the work, but the epigenome is going to be telling it what to do. So the hardware of your DNA is going to be the same throughout your entire life.
So again, the actual code, the ATCG, that stays the same. That doesn't move. The actual DNA stays the same.
But these epigenetic tags do change throughout your life, and they decide what genes get expressed or not. Now,
All right, so the rest of this is really great. It does talk about how more recently we're seeing more and more evidence that epigenetic tags, as they're referring to them, can be passed on to the next generation. But let's look at an example of those methyl groups in epigenetics and histone patterns. So before that, let's also clarify the histone patterns. The histones are the winding and loosening of the DNA. And if we go back one slide, here we have the DNA all wound up around a chromosome. If the DNA is more tightly wound, it can express less, or more loosely wound, it can express more. So that leads to it being different. So, right, if we go back to this slide, let's say this DNA is so tightly wound that it's so squished, the protein comes and it's expecting to read a certain thing and it's getting more information, right? Versus if it's really loose, the same protein might come in and get a different code, right? It might see a different number of letters. Maybe it's expecting, I don't know, a line of ATC, like 3 letters in a row. But because it's so tightly wound, it's getting 5 letters in a row. Or if it's too loosely wound, it's expecting 3 and it's getting 2 or something like that. Just as some examples here. So that is a histone pattern, the winding and loosening of the actual DNA. So a physical winding and twisting, making it tighter or looser. So the methyl groups kind of block off aspects of the DNA. So you can think of it like a big X right here, like don't read this part or don't read this part, versus the histones winding it up tighter or loosening it out. All right, so the other video in that slide is a really good example, has a couple really good examples, and it actually looks at Eevee as a Pokemon, which I think is fun because Eevee's one of my favorite. Eevee and, oh my goodness, why did it just leave my brain? Emolga, the like yellow lightning, like flying squirrel type Pokemon. Those are like my 2 favorite Pokemon. So this has a couple really neat examples in it. Feel free to watch the whole thing. It's got some good human examples as well as some good animal examples. But let's jump forward to the animal example of the honeybee. So it's right about here.
Their DNA that can be unlocked by certain triggers in their environment. A great example of this are honeybees. Yeah, I know. I just dedicated an entire episode to bee talk as it relates to Peachette. I'm just going through bee movie meme withdrawals. I don't know. Bees are fascinating creatures, and in this case, they are the perfect EV example. So, like I covered back in that episode, female bee larvae can grow into either a queen bee or a worker bee. What makes the difference here is the food that they're given at birth. Bees given royal jelly become a queen, while everyone else becomes a worker. So in this case, royal jelly is basically the evolutionary stone for bees, determining what form the larval bee evolves into. It even changes the bee physically, where, in their adult stage, queen bees and worker bees look way different. But
All right, so in this study, We have basically bees from the same hive, but you could clone a bee and do this exact same thing. So you've got a bee larva, they're two bee clones of the same exact bee. They have the exact same DNA strand. Epigenetics with the methyl groups can make the genes be read differently by the proteins in the body just based on some simple environmental factors. In humans, there's a lot more complexity in our DNA than, say, a bee, but we see some similar things. Our nutrition can affect our epigenetics. It can affect the methyl groups binding as well as the histone patterns. But for the bee, essentially, we have the two larvae exactly the same. In our case, they're clones. One is given a royal jelly, and the other is given like pollen, nectar, and honey as the food. So if they get the normal the poor bee food rather than the royal jelly, they will become a worker. Versus if they have the royal jelly, they'll become a queen. This is a really great example of the stark difference with those methyl groups binding to different spots on the bee's genome. Because again, our example, these are twin bees, these are or clone bees. Right? They are identically genetic, but they have different phenotypes. They look different. The queen acts different than a worker bee. So just because of royal jelly versus not royal jelly. So a really clear example of those methyl groups binding to someplace different. There's not that dramatic of examples in humans, at least not that we've seen, but that would probably be a little more obvious, kind of like this one. And next we can see the histone pattern with some mice, so the twisting and loosening. So in the first video they mentioned it's more like a knob versus this one's like a switch. So the methyl groups switch it to queen bee versus worker bee. The histone patterns are going to change things more gradually. We'll see a range of responses in the genome.
Bees and worker bees look way different, but in their larval stages, they're practically identical. It's exactly like two different evolutions that wind up with two different appearances despite starting with the same looking EVs. The queen and worker bees didn't start off as genetically different, but differences in their environment, royal jelly, or lack thereof, led to wildly different gene expressions. And bees aren't the only place in nature where this happens. Another EV-like example happens in the agouti mouse, which has a gene which changes the fur of its coat. These two mice are genetically very similar at birth, but due to different.
So while in this experiment, these were sibling mice, like from the same litter, you could do this exact same thing with all cloned mice. Like that's more expensive. So that's why they use siblings instead. But you could clone 20 mice. And so you have the identical genes for each of those 20 mice and see this happen because of the histone patterns. And it's in this case, it's again down to differences in nutrition, healthy eating versus not healthy eating in this case, basically.
Differences in nutrition, they have very different gene expressions, with one having a gene expression that causes it to become obese and have lighter fur. Same DNA sequence, different phenotypes.
All right, so I want to scroll back to this picture right here. There we go. So here in this picture, you can see the dark mouse over here, which is kind of more standard, what you would expect. And then with unhealthy nutrition, a lot more obese and a lot blonder fur. This is the example of that histone pattern twisting and loosening the DNA. So the genes are kind of expressing how you'd expect and different and more different. So this is a really concrete example of those histone patterns and the winding and loosening or the tightening and loosening of the DNA. Again, that protein is going in to read the DNA, to ask it what its instructions are in the cell, and this is the outcome. So this happens in humans as well. So humans also are changed by epigenetics. We have the chemicals that we eat, things that we're breathing, smoking, the sleep or lack of sleep that we're getting, the increased stress hormones in our body. All of these can change methyl groups, histone patterns to lead the proteins to do something different in a cell. And so these chemical process compounds that make up the epigenome attach to genes, the methyl groups turn it on or off, or they help alter it with the histone patterns to make it happen more or less. And we can alter these through different things in our environment. So again, this shows us these gene environment interactions are very complicated. For any given thing, there's multiple genes and they're being affected by the epigenetics of the gene, your nutrition, your sleep, And generally, what we see is the things you already kind of know to be healthy. So, eating a healthy diet, getting regular exercise, sleeping well, these things all help our genome perform as expected or optimally. Versus, we're seeing more evidence that if we are not doing that, our epigenome is um, a bigger risk factor. So, with this, it can show how wildly different behaviors can lead to different gene expressions. So, this is another example of this developmental cascade. You might have a specific gene, but based on your environment, it might be more of a protective factor or more of a risk factor. Then you change your behavior. You change your environment. You change your diet. You change your sleep patterns. And maybe you have more protective factors, or maybe you do the opposite of that. You're staying up a lot more. You're not getting enough sleep. It might push you back towards more risk factors. So genes and environment are always working together. Like so many things, development is genes and environment affecting each other. And a big piece of this is also you and your individual choices. What you do, what you put into your body, how you take care of it can literally affect the way those proteins come in, read the DNA sequence, and then go and do something in your cell. So it's a really cool area of study that we're learning a lot more about. Most of the studies in this area have looked at XYZ negative thing in humans. Does epigenetics explain it? And very often there is some epigenetic component to a lot of these things. So a lot of the studies we're finding now are kind of negative things that epigenetics happens, but also it shows that, well, if you're eating poorly and it's bad for your epigenetics, if you eat well, it can be good for your epigenetics and your genes will do better things in your cells. Your cells will be more happy and healthy body overall. Another thing with epigenetics, it does seem like some epigenetic information can be passed down. It's not always, and not everything is passed down, but there is more and more evidence that we can pass on some of that epigenetic information, like where to bind the methyl groups, how tight or loose to wind the DNA strand. So, interestingly, our nutrition choices can also affect a child that we would have. Obviously, if you're already done having children, then your epigenetic changes aren't going to affect your children anymore. It's just before the reproduction part of it. Another big part of this chapter is the nervous system. So this includes our brain, which includes billions of cells, neurons, glial cells, and these are what we usually refer to as brain cells, is the neurons. Usually we're referring just to the neurons. We also have sensory and motor nerves all throughout our body. So if I reach out and touch something that's hot, I've got a microphone out here. I'm not going to touch it because that would be loud. But if I did touch it and it was hot for some reason, my sensory neurons would tell me it's hot, and my motor neurons would pull it back. Our spinal cord is basically a bunch of axons. There are neurons in there as well, but the long component sending messages back and forth is what the spinal cord is largely made up of. And then we also have our endocrine system. So the endocrine system is our hormones in our body. So our stress hormones, the hormones that tell us to calm down and go to sleep. All of these are also part of our body's electrochemical communication circuitry. So the neurons are usually what we focus on, but the hormones in our body are also a big part of that. So, with this, there's a couple key things to know as we're talking about the nervous system. One is specifically about the brain's ability to be plastic or plasticity. Plasticity basically means the capacity for change. So, when I say plasticity, think about things moving or modifying. Think of like clay or Play-Doh, plastic that can bend and not break. So, the brain has capacity to learn new things. Most of that happens because of dendrite and axon branches, which we'll get to, or hopefully you've read in your book already. So a lot of this is just a physical connection of new axon branches to new dendrites to make new networks. The reason I have a picture of a taxi in London is because there was a really interesting study done. It looked at taxi drivers' brains versus Londoners' brains, and it found that in the areas of our brains where we kind of store our mental maps, so how do I get home from work? Where are things in my house? Like that kind of mental map. The taxi drivers' areas of their brains were so much more dense. There was so much more material there in their brains, and then the Londoners who might have been their neighbors. Right? And so it was something that was measurable. We could see much more mass in their brains where those mental maps were. And essentially, this comes from practice over time. They learn the roots. They're doing it every day. They're practicing it a lot. So there's a lot more dendrites and axon branches, so many more that it was really easy to measure. So that's plasticity. We can teach an old human new tricks, essentially, is the idea there. Another big key is integration. The brain is doing a lot of stuff right now. You are hearing things, you are seeing things. There might be smells around you. You might be cold or hot. Your stomach might be hungry or not hungry. You might need to use the bathroom. The brain is integrating all of that information to tell you what's important right now. What do you need to focus on? As you're driving, hopefully it helps you focus on the dog that runs out in front of you, right? It's integrating all the information and helping you make meaning and make connections and focus on what is the most important in any given moment. So now that we know that the brain is relatively plastic, that does this integration, let's talk about a couple key components of the body. So one is afferent nerves. So these are the sensory nerves. They're carrying information in. So again, the afferent nerve would be giving the message that, oh, this microphone is really hot for some reason. Efferent nerves carry messages out, so to the muscles. So my brain telling my arm to move away from the hot thing, that is going through efferent nerves that then talk to muscles. So the efferent nerve is connected to muscle cells saying contract, contract, contract. All right, so that's afferent and efferent. Those are part of the nervous system, but it's not our usual focus. In chapter 4, there are specialized sensory nerves like rods and cones in your eyes. So that's another thing we'll talk about later on. Glial cells are additional cells. It's basically a category of brain cells that are not the neurons. There are a bunch of different kinds of cells in your brain that do a bunch of different jobs, and we kind of give them all the name glial cells, and up until recently we didn't really care about them because we didn't think they were that important. We thought the neurons, and I guess you could make the argument that the neurons are kind of the most important for our brain processes, but they couldn't do their job without the glial cells. The glial cells make insulation on the axons of the neurons. So they make the myelin sheath to make the message faster. They do help with some of the communication. Some glial cells make some of the neurotransmitters that we use. They can reorganize and put neurotransmitters where they're supposed to be. They can be part of signaling as well. And that's some things that we've discovered more recently. They also do a lot of nutrient and waste transport. The neuron is a cell. It needs energy. It has waste. It does normal cell things. So it needs to be supported with blood, with nutrients, with vitamins, minerals, the kinds of building blocks of proteins and things like that. And it also has waste that has to be taken away. So green on this image here is a neuron connected to a bunch of other neurons, and then the red cells are the glial cells supporting that neuron in doing its job and supporting other neurons in doing their job. All right, so here is a neuron, a little image. Some of this is probably blocked by my face, depending on where my face is on the screen right now. But up here, we've got dendrites, then we've got a cell body, we've got an axon, we've got axon branches, and then we've got a myelin sheath. So with this, all neurons have some of these same basic characteristics. And so let's talk about them. A cell body is the same as other cell bodies. It directs the growth and maintenance of this cell. And that's a key thing about the brain. These neurons are cells. They take energy. Our brain uses, I forget what percentage, like a third of the energy and nutrition that we give our body. It's a very energy intensive organ. And the nucleus and the cell body direct that. There's mitochondria in there, the DNA's in there. And so that is the cell body. It's a very important part of keeping that neuron itself alive. Even though usually what we talk about is the axon, sending messages. It's like the message delivery highway or like the main wire sending messages. So here is the axon. Neurons only have one axon, but that's a little misleading because there's a lot of axon branches and you can grow new axon branches. So even though there's one primary axon, there can be multiple branches connected to up to 1,000 neurons in general, both on the back end at the dendrites and at the front end. Total can be connected to as many as 1,000 other neurons. So there can be lots of axon branches. And in humans, our longest axons are in our spine. They go from our brainstem down to our tailbone, basically, and they can be up to 3 feet long, which is pretty neat. Giraffes have some that are like 3 meters long, which is very long. Basically the same, like all the way down their spine. All right, so with this, dendrites are, you could think of them as the antenna. They're receiving information, taking it into the cell, and then down the axon. Most cells have many dendrites. A lot of plasticity is the dendrites getting more messages from more places, so you can make a message highway from a cell down this pathway to this axon, down this axon branch. So that's one pathway. Then there's another pathway for another process in your brain that goes from this dendrite down this axon branch to another set of cells. So that's the dendrite, they're receiving messages. Then the myelin sheath is a really important piece. It insulates the axon, and that essentially speeds it up. So human neurons on average are refreshing or able to send a message 200 times a second. They can go faster. Like the theoretical maximum is like 10,000 times a second. But in measuring, we usually see like 600 times a second as the fastest that humans do. But again, the cells like to use less energy. It takes energy to send messages, so it wants to be efficient. Most human neurons are going like 200 times a second. So let's watch a quick little clip about a human stem cell becoming a neuron. This is how we get all of our neurons. No neurons divide in and of themselves to make new neurons. The only thing that divides is the stem cells, which can then become neurons. Some of our other cells in our bodies can divide, but neurons specifically do not divide to make new neurons. So basically, we've got all that we have when we're about 2. A lot of neurogenesis, the stem cell becoming a neuron, happens when we are prenatal, and then up until about age 2, we still have stem cells becoming neurons. And there's a couple places in our brains that still do it even into adulthood, but it doesn't seem like it has an impact, those couple little areas in our brain. That make new neurons. So let's watch this. There's a key piece here. I might actually just flip. Feel free to click through and watch this whole video. It's pretty short, like 2 or 3 minutes long, as well as some of these on this page are really interesting. But I really want to focus on kind of the last step here, starting about here. So these are stem cells becoming neurons. So essentially how a stem cell, oh, that was far louder than I thought. So stem cells, as we see here, can become any cell in the body. Basically, it has to get its instruction about where the methyl group should go. So there's neurogenic cell medium that essentially our bodies produce and tells these cells, hey, you're in the brain, become a brain cell or become a neuron. So that's what's happened. They have the information to become brain cells, and let's see how quickly it does. Keep track of this time in the corner as well. So it'll zoom in on a specific area. A lot of these cells are trying to become neurons. And we can see this stem cell is pretty far into the process, retracting its cytoskeleton, and now we see the stem cell is a neuron. So you've got the cell body, you've got some dendrites, you've got an axon, and the next step in this process will be it'll reach out and try to communicate with other neurons. So if you want to watch that back, stem cells can become, oh, here we've got a neuron. We've got some dendrites. This thicker one is probably the axon in this example. But so this one is already a neuron, trying to make connections with things all around. And then I'm not sure if there are glial cells in here. I'd be surprised if there's not also glial cells. I think it started off all as basically stem cells in a petri dish kind of thing. But yeah, that is a neuron, and it can pretty quickly, a stem cell within like 8 to 18 hours, become a neuron rather than a stem cell. From that, it might also let you know that we can do this process of growing new dendrites or growing new axon branches pretty quickly. It can take a couple hours, like up to like 6, 12 hours. And that's why things like learning can happen so quickly. You can make brand new connections. You can make brand new dendrites and brand new axon branches that connect up and have a new pathway in the time it takes to sleep. And a lot of that is done during the time you are sleeping as well. It's not all done during sleep time, but it's a big time that it is happening. All right, so the communications between the neurons happen at the synapse. It's a little gap between the neurons. They're not really touching. They are basically, here's the neuron 1 that's delivering a message, and at the end of this synapse, it has neurotransmitters. So it spits out some serotonin, maybe some dopamine, and that gets into basically a gate, little molecular gates that open up the next two ions and start a message. And we'll see in a moment the process by which that happens. So the receptors are on the other side. They receive the message and then start a electrochemical process in the next cell. So let's watch the majority of this video for some information. For more chemistry in here, watch this video. So if you're going into like neurobiology or chemistry, this one might not be too advanced for you. It might be great and connect with some of the things you already know. This one already has more chemistry than we need for this class. So I'll point out some of the more specific things here that you need to know as you are learning about how neurons work.
Pain you experience when you burn your hand results so quickly in an action by your muscles. Many animals respond to environmental stimuli using specialized cells called neurons. A stimulus is detected by sensory receptors, and the body responds through motor effectors. These cells working together allow you to respond very quickly to threats. When you touch something hot, heat receptors of a sensory neuron detect the stimuli and send the information of heat to an interneuron in your central nervous system. From there, a motor neuron sends a response from your central nervous system to the skeletal muscles in your arm, causing them to contract and pull your hand away.
So here we've got a couple different types of neurons. We've got afferent and efferent neurons. We've got the neurons taking in the information to the brain stem. So here the spinal cord or the brain have interneurons. Then the motor neuron sending a message back out to the muscle cells. But you can see each of these has a cell body. Right? Each of these has some dendrites. You could think of the sensory components like your rods and cones or the specific sensory cells in your skin as a specialized type of dendrite. You could kind of think of it that way. And then it's sending the information to the next neuron in the chain, the next neuron in the chain, and then at the end, rather than spitting out at the synapse those neurotransmitters, it's spitting out the chemicals that tell a muscle cell basically contract. You've probably seen something like that if you've ever, say, filleted a fish that was really, really fresh. If you pour salt on it, that is a chemical that can make the muscles contract. So essentially, we're just talking about molecules being manipulated to make the brain and body do things. All right, so we're going to zoom in to an axon to see what's happening at the molecular level of an axon.
The fundamental process of neural transmission that underlies this action occurs in all neurons of the body. Neurons transmit this information through changes in the electrical potential of the membrane.
All right, so again, there is a lot of information in this video that you don't necessarily need to take with you in this class, but it is a really good demonstration of these processes. As a clarification, here we've got one of the membranes of the axon. This down here is the inside of the axon. And then below the screen, we have another membrane of the axon. So this is the inside middle portion. There's another membrane somewhere down here out of frame with gates on it as well. Out here, we have the rest of the brain. You can see some other neurons in the background kind of floating around in a soup of brain. So here we've got a positive charge on the outside, a negative charge on the inside. This is the resting potential of an axon. And so we'll see a charge go down, but we're not actually shooting any of these ions down the axon. So let's see how this happens.
By the movement of ions across the membrane. An electrochemical gradient governs the movement of these ions, resulting in an electrical impulse. The resting membrane potential in a neuron, when the cell is not firing an impulse, is established by the unequal distribution of sodium ions outside of the cell and potassium ions inside the cell. making the outside of the cell more positively charged compared to the inside.
All right, so ions are simply a charged particle. Right? It is a little molecule that has a positive charge. So I've got the sodium ions out here, and there's enough of them that there's a positive charge. We've got some potassium ions in here, and then there's a little bit less of them in here to make a negative charge. So slightly positive, slightly negative. And what we're going to see is these ions, they don't go zip all the way down the axon. They don't go zip all the way down a 3-foot axon. They go just a very minuscule amount. They go back and forth across this membrane. And that is what changes the charge. And that change of charge leads to a chain reaction down the axon.
The electrochemical gradient is established and maintained by an enzyme called sodium potassium ATPase. When a neuron is...
So if you never remember sodium potassium ATPase, that's fine for this class. Like there are some chemical compounds that we don't have to memorize for the class. Some of them it is useful, like knowing that sodium is on the outside and potassium is on the inside. But all the video has a little more information than you need to know, but it is a good demonstration. So now we see the charge indicated by the glow. Probably be better if they had that glow kind of in here, but it's a pretty good visual demonstration of what's going on.
Stimulated, sodium ion channels open and sodium ions flow into the cell. This leads to a change in the electrical potential across the membrane called depolarization. The depolarizing electrical potential travels down the dendrites and over the cell body. Multiple electrical potentials will combine at the axon hillock in a process called summation. If the depolarization is large enough, an action potential is triggered. Action potentials are all or none electrical impulses that maintain their amplitude and strength down the length of the axon. The action potential travels down the axon when the depolarization of an area of membrane causes adjacent voltage-gated sodium ion channels to open.
So to repeat that in just more plain language, this charge, as the ions come in here and it gets more positively charged, it opens up the next gate. So the positive charge tells the gate to open, which then in turn makes it more positive in here, which in turn opens the next gate. So it is a chain reaction. Gate open, gate open, gate open, gate open, all the way down the axon. And so in that process, the ions are only moving across the membrane. They're not going down, but the resulting charge is same strength the whole time passing down through the axon.
Influx of sodium ions results in membrane depolarization along the membrane. After a short delay, potassium ion channels open and potassium ions flow out.
So again, all these processes are taking some energy to move them against the concentration gradient. And so the neuron is using energy to do that. And also the short delay, it's a very short delay because again, neurons can fire like 200 times a second.
Repolarizing the membrane. For the neuron to fire again, the resting membrane potential needs to be reestablished. Sodium potassium ATPase is used to move sodium and potassium ions against their concentration gradients, reestablishing the resting membrane potential.
So here we've got a good diagram. This is the axon. This is the top membrane. This is the bottom membrane. This is the inside of the axon. This is the outside of the axon. And we will see the chain reaction of it getting a little positive, opening the next gate, getting positive, opening the next gate. And again, that happens very, very quickly.
As the action potential moves down the axon, ions are diffusing only a short distance, allowing the signal to move quickly. At the axon.
So hopefully now you have a lot clearer picture of how that electrical charge is going through. Again, it is not the ions shooting the whole way down the axon. That could fail more often than this, right? That might lose its strength. It might bounce into something wrong. This is the charge is moving. The charge is chain reaction moving all the way down the axon. Then we'll see what happens at the synapse, how we then talk to the next neuron.
And terminal. The electrical impulse passes to another cell at a cellular connection called a synapse. The space between the presynaptic neuron and a postsynaptic cell is called the synaptic cleft. The presynaptic neuron contains signal molecules called neurotransmitters, that are packaged inside vesicles. When an action potential reaches the end of a neuron, neurotransmitters are released by exocytosis from the neuron into the synaptic cleft. Neurotransmitters bind to the adjacent cell at receptor sites attached to ion channels. The channels open, allowing the movement of ions into or out of the effector cell, which alters its membrane potential, thereby transmitting the signal from the neuron to the effector cell.
All right, so let's go back to this image. So basically what's happening, that charge gets to the end here, the vesicles then move to the edge past the membrane, spit out some neurotransmitters, which are chemicals we'll cover in just a minute. Those chemicals attach to gates on the next cell and open it up for the ions to pass through, which means the message continues on in that next cell. So the electrical charge passes through the axon to the end, to the synapse, spits out some neurotransmitters. The transmitters connect with the next cell in line and let that cell continue a message. So a good example of a neurotransmitter is dopamine or serotonin. These might be ones that you've heard of before. They get spit out, connect over here, send the message to continue on that path. And we'll talk about the different functions of a couple different types of neurotransmitters in a few minutes. But yeah, hopefully that made it a lot more clear. Again, more chemical focused and chemistry focused video here. And that action potential is just what we describe that electrical charge and the pattern that it is. The reuptake is the revacuuming up essentially of those neurotransmitters so they can be used again. Resting potential, again, is just that state of readiness of the neuron. And this is all important because that message sending at the synapse, that message sending through the neuron is an essential piece of how we think about things, how we remember things, how we love, how we perceive, how we experience or create and live. These basic biological functions at the molecular level are how we experience life, how we are doing things, how you're listening right now or interpreting it or asking questions. And so this is essential for all other aspects of psychology. So that's why we dive into it in this much depth in a general psychology course, because we'll come back to the brain and the neurons themselves as the cause of a lot of things that we see later on down the line and how we can leverage how our brain works, how these neurons work to do things like learn or build good habits and things like that. So with that, we will jump into some specific neurotransmitters. Neurotransmitters are the specific chemicals that we send as signal messengers across the neurons and across those synapses. There are 100 specific neurotransmitters, but they're all basically different variations of basically 8 basic neurotransmitters. So those 8 neurotransmitters and 100 specific neurotransmitters have 3 primary jobs, and those are either excitatory responses to the next cell, inhibitory responses to the next cell, or modulatory responses to the cells around them. So you can think of excitatory responses as do the thing or send the message. Inhibitory responses are like stop the message, don't activate anymore. And the modulatory is just adjusting or modulating the message to a group of neurons. So the main categories of neurochemicals or neurotransmitters are acetylcholine, GABA, glutamate, norepinephrine, dopamine, serotonin, endorphins, and oxytocin. So those 8 neurotransmitters are the building blocks of basically all of the messages across our neurons. Here's a little picture and some graphs of the types of neurotransmitters, a little bit about the effects that they have, but we'll go into a couple of these in a little more detail. But feel free to refer to this slide or the other slides for more detail. We'll only focus on a couple to give you an idea of how these work, but definitely review this information and review in your book to see a little more information on neurotransmitters. So first example is acetylcholine. Acetylcholine stimulates neurons to fire. It's relevant to moving muscles, your memory, to create a memory, learning new things. All of that is part of what acetylcholine does. So each neurotransmitter can do a lot of things. Again, we have those 8 basic ones and variations on the molecule. It can make it like a slightly different form of acetylcholine and things like that. But with this neurotransmitter does that excitatory thing. It tells muscles to fire, or not muscles. It can be involved in muscles, but it tells neurons to fire. It's exciting responses. So Alzheimer's disease is partially caused by acetylcholine deficiency. So that's part of it, that you're not getting neurons to fire that should be firing for, say, a memory. But also Alzheimer's does kill neurons over time too. So if the neuron is totally gone, well, then there's not a lot you can do. But there are different medicines that can work and help with acetylcholine. Um, there's, other than keeping your brain healthy in general and your body healthy in general, there's not a lot you can do about the death of those neurons. So that piece will still happen. That being said, as neurons die off, the plasticity, the dendrites and the axon branches, they can compensate to an extent for losses. So it can decrease the decline that you're experiencing up to a point. So that is one specific type of neurotransmitter, the acetylcholine. GABA is really common, used in a lot of things in your brain. So it in combination. So any given process that your neurons are sending, let's say the message gets to the end of the synapse, and so you're sending out, let's say, 50% GABA, 50% acetylcholine to send a specific message down a specific network of the neuron you're sending it to. So this is preventing neurons from firing in specific combinations. So a specific chemical code of how much acetylcholine or GABA or dopamine all together tells the next neuron kind of what network, what it's supposed to be doing. So, but again, we're only going to go in detail about a couple of these. This has a couple different variations of chemical GABA right here. Or maybe that's all the same, GABA right there, but a couple different chemicals that are similar. All right, one that I want to focus on, because you've probably heard of it, is dopamine. So dopamine is an excitatory one again. It is related to a bunch of different things with our personality, our mood, our sleep, attention, learning. And so dopamine is used in a lot of different processes. Dopamine is one that cocaine and amphetamines basically pretend to be and activate dopamine receptors. So you remember at that synapse, the second component, so where it's the neurotransmitter connects to a gate to start the charge of the next neuron. Cocaine, cocaine, cocaine and amphetamines essentially pretend to be dopamine. Their chemical structure is similar enough that they bind to those receptors and have the neuron do things, even though there's not dopamine there. So another one that you've probably heard of before is serotonin. Serotonin, again, does a lot of things. Serotonin regulates sleep. It helps with mood, but also attention, learning. So you can see some of these excitatory ones are involved in learning, things like memory. So it works with acetylcholine and norepinephrine very often. So again, the previous neuron is putting out a specific combination of serotonin, acetylcholine, norepinephrine to tell the following neuron exactly what kind of network you should be continuing on with the message. So it's very important to blame brain plasticity. So with this, some antidepressants, some of the most common antidepressants, affect the reuptake or that revacuuming of the serotonin and try to leave it in that gap. So you just already have more serotonin in the gaps between the neurons. And then if you have more there, it's going to be more likely when it's spit out by the preceding neuron that some connects with the next cell. So this is what SSRIs are. Selective serotonin reuptake inhibitors. So it's inhibiting the reuptake of serotonin. So it keeps it there in the synapse. So when the cell A spits out some more serotonin, there's already more there in the synapse. So it's a lot more likely that the next cell will get some serotonin connected with it and send that message. So that is the idea. And this is also why a lot of people who have, say, the same diagnosis, whether it be depression, anxiety, ADHD, whatever it might be, might need to use a very different medicine from their sibling or their friend or their partner that has the same disorder. Oftentimes, doctors will basically start with the ones that are the cheapest or that your insurance covers. And so SSRIs are a really common one for that. Also ones with less side effects too. And they'll basically run down. We'll use this one on serotonin because it keeps it in the synaptic cleft. We'll use this one for serotonin because it makes you produce more of serotonin. We'll use this one that does serotonin and dopamine. And you'll basically have to do a trial and error to see exactly what does your body need to feel your best and do your best. So that's kind of why oftentimes you have to do a trial and error with these types of medicines. Because even though you have depression, for example, it might not be serotonin for you, right? That might be a piece of the puzzle, but maybe it's also dopamine or other neurotransmitters and things like that. So that's why it can be hard to figure out exactly what kind of neurotransmitters you need to influence. Even doing a bunch of medicines sometimes doesn't work for some people. So there's other treatments as well if that is the case. With that being said, though, hopefully in the future we can get a lot better measuring tools to see how your neurotransmitters are in your individual brains. It's not an easy process for what we do right now. All right, so those are the couple that I really want to focus on because the main thing I want you to know about these neurotransmitters is, as well as these basic facts about them, know that some of them are excitatory, some are inhibitory, and some are modulatory, and they're working in combination together to get a message from cell A to cell B to do all the things that your brain does. So spend some time and look over all of these in the slides, or pause the video and read all of them. I'll go back to the beginning so you can go through, pause the video, read each of these. We talked about acetylcholine. We only mentioned GABA, glutamate, norepinephrine, dopamine, serotonin. We did talk both about dopamine and serotonin. Endorphins are an example of a chemical that's both a neurotransmitter as well as a hormone. Oxytocin is another one that is both a hormone as well as a neurotransmitter. And that's basically just saying it's a chemical that sends a message in your neurons or a chemical that tells your organs things to do. So oxytocin, just a molecule, just a chemical that can be read by the next neuron, open up a gate, send a message, or by organs in your body. All right, with that, let's look at a specific kind of neuron that's pretty neat. It's called a mirror neuron, and it really shows some of the capacities of our brain. I think this is a really fun story about how some neurons called mirror neurons work.
If you read some of the less reputable corners of the internet, you might have heard strange things about mirror neurons. According to some people, they are why we have empathy and ESP and telepathy. It probably goes without saying that most of that isn't true. They definitely don't give us superpowers, and there is conflicting research about whether they're the basis of empathy. Even if they're not, though, they are still a very cool part of your brain, and probably they help us learn from other people. The name mirror neuron comes from the fact that they fire both when you do something and when you watch someone doing something, even if you don't move a muscle. Basically, it's as if they mirror the neural activity of the person actually doing the thing. And they were first discovered in macaque monkeys in the early 90s. The story goes that one day, while a monkey had thin needle electrodes inserted into individual neurons in their brain, some of the graduate students, well, they went and took a lunch break. And while one of those graduate students was eating in front of the monkey, all of a sudden, there was neural activity in the premotor region of the frontal cortex in the monkey's brain. This region would normally fire if the monkey was moving, like reaching for food or eating. But those neurons were firing while the monkey was motionless and just watching someone else do those things. Further research started mapping out exactly where these neurons were, and they're remarkably specific. Like mirror neurons that fire when an experimenter grabs a piece of food wouldn't fire if the experimenter used tools to grab the food instead. But the big question was whether this happened in humans too, because these neurons were in regions of the brain where monkeys and humans differ quite a lot. Of course, your average human study participant is a little fussier about getting electrodes planted into individual neurons inside of their brains, so scientists used electroencephalograms, or EEGs, to look for mirror neurons instead, as they record neural activity through the scalp. Researchers had 20 subjects watch an experimenter demonstrate some motions with their thumb and index finger, and then perform the same motions themselves. And turns out some regions of their brains did act like the monkey's mirror neurons. Parts of the motor and premotor areas of the frontal lobe, for example, as well as where the parietal lobe meets the temporal lobe, an area involved in vision and focusing attention. attention. But
So I'm going to pause it right there. A little bit later than I meant to. So this, you're paying attention, right? You're watching someone do an action, so you're focused on that action. When you're doing that as a human, and some primates do this as well, so non-human animals, as you're watching a human or even something similar to a human, like a human model, or even in this example, the ape or the chimp, or yeah, the monkey. I think they're all different categories. The monkey was watching a human do it, and their motor and premotor cortex were active. So essentially, this is where you're kind of planning out an action. Like, I'm about to do this action. Like, the intent to do an action is also coming from the frontal lobe or the prefrontal cortex. And then it's like, all right, prep the action. And then this says, do the action. So left side of my brain, right about here, is telling my right hand to move like this. And now the right side of my brain, right about here, is telling my left hand to do this. So we've got that bilateral thing. The left side of your brain is getting the sensory input and directing the right side of your body. The right side of your brain is getting the sensory input and directing the left side of your body. So the vision going through my left eye right now is going to my occipital lobe on the right side, way back here. So pretty neat. But anyway, the mirror neurons are there in the areas for doing a motion. So essentially you are watching someone do it, and your brain is kind of pre-running that motion. So you're not actually moving, but your brain is kind of doing some of the same neurons that would be active if you did the motion. It's like it's doing it on its own. It's practicing the motion before you actually practice the motion. And we've seen, and we'll see from the results of this study, that it leads to better performance in humans.
Example, as well as where the parietal lobe meets the temporal lobe, an area involved in vision and focusing attention. But knowing that they're there and where they are doesn't tell us why they're there. Many neuroscientists now think that they help us learn from other people through observation. Take a 2009 study, for example, which looked at how these parts of the brain are active while people were being taught how to dance. The researchers used a game similar to Dance Dance Revolution and varied whether the participants were taught the steps using symbols on a screen or a human model in addition to the symbols. And they found that parts of the mirror neuron network really tune into human actions. For example, the superior temporal cortex was more active while watching a person demonstrate the dance than the symbolic breakdown of the steps. But perhaps more to the point, the participants' performance on the final dancing test was better when they had a human to watch, which suggests that these regions support observational learning. Human mirror neurons aren't exactly the same as the monkey ones, though. Ours seem to care about the actual movement, while theirs are more strongly tied to the movement's meaning. Like, say you grab a water bottle the way you normally would, and then you put it down and grab it with your hand like upside down instead, which makes drinking it a lot harder. It's basically the same muscle motion either way, but one serves a meaningful function. In a macaque, watching the meaningless action might not activate their mirror neurons. But in people, the mirror neuron regions of the frontal lobe activate regardless of whether the action makes sense. All that matters is the actual hand motion. It's possible that this helps explain why humans are so good at learning by imitation. Part of our brain is always paying attention to motions that seem meaningless, just in case we can learn from them.
So that's a big key with these mirror neurons. And one thing that does take a lot of energy for our brain, because again, these neurons need energy, they need to maintain themselves, they need steady streams of things that cells need, right? So fuel, they need their waste taken away, and it takes energy to do that. So part of our brain is always watching for potentially learning opportunities from watching someone else do something. So as you watch, I am lifting up this mug. It might go away with the green screen. But as I lift it up, your brain is also doing that. So you are pre-running that motion because it might be useful. Even if I hold it a really weird way, you might look at that and say, oh, there might be something that I can learn from this. This is part of the reason why humans are really good at observational learning. It also is connected with if you intentionally practice, like mentally practice. So you imagine yourself doing a thing. So we've seen that for athletes, we've tested a lot of this in, say, like college or Olympic athletes, watching their own performance and then from like a play or a gymnastics move or whatever it is, and then themselves imagining what they would do differently, then they perform better than if they just practice more. So the mirror neurons of seeing themselves do it, see maybe other examples of them doing it correctly, and then also visualizing themselves do it, that all influences your motor cortex. It's active in a lot of those processes, making it easier. It's another good form of practice. I think one study was even with like people who didn't really play darts, and even just like visualizing and seeing themselves play, they did better, like with the mirror neurons plus the visualization. So all these components together can lead to us learning, and it really all goes to show that we have a lot of capacity for plasticity. We can change and modify our brain in really amazing ways. So, and basically a lot of this is the physical process of connecting between neurons. So we've got these dendrites, we've got these axon branches, and basically a cell just needs to grow a new dendrite branch, grow a new axon branch, and make more connections. It can also make the connection stronger. If you go to the neurobiology class, we can talk about it, but basically the more you use a network, the easier it is for the neuron to use that same network. So that's why habits are hard to break, because it's very low energy. The neurons kind of learned, hey, I can do this. This is important. I do this all the time, so I'm gonna do it with less energy now. Versus something new takes more energy to learn. So this kind of plasticity mainly happens in three specific ways: collateral sprouting, substitution of function, and neurogenesis. So I'll actually jump forward and start with neurogenesis. So neurogenesis is the process of new neurons being created. As we've talked about, this happens mostly in prenatally and up to age 2. We've got a lot of stem cells becoming literally all of the cells of your body. Your stem cells are becoming every single organ in your body. But those stem cells are also becoming the neurons, the brain cells. And so they migrate to where they live, they start making connections, that kind of thing. But once you get to be about 2, that neurogenesis pretty much stops. So brain plasticity in human is not really down to neurogenesis after about age 2. There are these two specific zones, the SVZ subventricle zone and the olfactory bulb. Both of those have stem cells becoming neurons still, but it's in such a small amount, it probably isn't making a difference. There are some interesting studies that show that in some cases the olfactory bulb new neurons might be making smell and taste a little bit better into later adulthood, but it's not for sure. Because basically it's a very small amount of new neurons being created. So the main ones that humans do are collateral sprouting and substitution of function. Collateral sprouting is basically in your brain, you've got a network. Let's use the gymnastics example. When you're going to do that gymnastics move, you're using your eyes, you're using your balance, you're using your motor cortex, you're using your frontal lobe to plan out how you're going to do this. So there's a lot of areas involved in that function of doing this vault, let's say, in gymnastics. So if you say have damage, whether it is from a seizure, a stroke, traumatic brain injury, whatever it might be, you might just have a neuron die. If you have alcohol poisoning, you probably have neurons die. But if some of the neurons in that pathway to do this vault in gymnastics are damaged, there's others that are part of that pathway that can kind of take over the responsibility of those now damaged or dead neurons. So usually this is nearby cells. So cells that are in a similar pathway or were part of the same pathway kind of take on that function. So that's collateral sprouting. You make new networks kind of around, you could think of it as going around the dead ones within the same network. So that's one of the main ways we do it, and that's a lot quicker for us. That's just building some new dendrites, building some new axon branches, and making those networks stronger for this thing that we need to do, the gymnastics vault. Substitution of function is a little more intensive. Essentially, it's taking a brain region that's maybe too damaged or has too widespread damage to do that collateral sprouting, and it moves those functions to another part of the brain. We do this better when we are younger and not as well as we are older, but it is still possible. So language, for almost all people, even things like sign language, is processed here on the left side of your brain in this temporal lobe. So kind of like right above your ear, like right about here. That's where most people's kind of language centers are. But if you have that damaged, other parts of your brain are part of speech, right? You have to literally move your mouth and tongue, right? You have to be paying attention to other people. So there are other places in the brain that are part of that network, even if they're not close to that network. So usually it's some part nearby or some part of that network, even if it's on the whole other side of the brain, that can substitute and take on those functions. So that substitution of function, these are really dramatic examples of brain plasticity. So this usually happens, again, in places that were part of the pathway, part of the process, but this is like a really big reorganization. So your language functions move to this side of your brain. And so those neurons are now pulling kind of like double duty. So human brains are really amazingly capable of plasticity. There have been cases of people who go in for an MRI for whatever it might be. Maybe they have a headache and they're not sure why. And they're getting an MRI to get diagnosed and they realize, oh my goodness, you have half of your brain. This has happened that people have realized, oh, I only have the right half of my brain or the left half of my brain, just at random. But their whole life, their intelligence seemed the same, their personality seemed the same as everybody, because that half of the brain is doing all of the functions. So this also goes to the myth that we use 10% of our brain. At any given moment, we're probably using closer to like 60, 70% of our brain with doing things, really intensive things that we're using all of our body and muscles. We might get higher, but we're always using a bunch of different networks. So it's hard to put like a 10% number to it, but it's probably closer to like 60 or 70. And what that means too is that the brain is really capable of doing a lot, moving these functions, substituting the functions, also the collateral sprouting to take on functions. So it is really amazing. So this is a video that for some of the classes might be part of your hybrid activity. So I'm going to have it here. I'm going to make some points while the video is playing, and then we'll go on to basically how we study the brain and a couple specific brain areas. But if I mentioned for you to watch this separately, go and like open the video and just watch it separately. But I'm going to play it through right here and make some commentary throughout as well. So I tried to upload the lecture video with this video playing. It will not work. It is copyright material, and so even though this does fall under free use to share it during class, it's going to be a process to get it to actually play with the lecture video. So instead, go and watch this video separately. So click on the link through the slides, navigate to this through this website, or depending if you're in an online class or in-person class, you can either find it in the slides or in like the video instructions for the week. So click that link, watch the video, and then in the next couple seconds, I will be back talking about what was in the video and relating it to the neurons. One thing that you miss out with that is my commentary throughout the video. Some of the main things are pay attention to the side of brain that was injured, some of the components that were probably injured in Richard's brain throughout this video, and some of the observations of the kinds of training that they were doing to help the brain in its process towards plasticity. So sorry this isn't part of the lecture. You're going to have to navigate away and link and click on that link wherever it is for your class. But yep, I'm not able to keep it within this course video up on YouTube. So really dramatic example of brain plasticity. Here we see examples of both that substitution of function as well as that collateral sprouting, and it's really impressive what his brain was able to do. Really dramatic example there of what the brain is capable of. So with that, let's move on to how we study these different parts of the brain and the specific areas of the brain. So as you're looking at this next section of the chapter, consider Richard or other people in your life and areas that they might have had damaged in their brain and think about how these different areas of the brain are relevant to that. So let's look next at how we study the brain and different areas of the brain. So one of the ways that we studied the brain before we had the advanced tools that we have now is called brain lesioning. Essentially, we would look at the abnormal tissue or damaged tissue from disease after a person had died. So after they died, we would look at their brains, see if this explains or if we could find patterns of what explains specific behavioral outcomes. So you would have a person who had some sort of difference of their personality or difference of their vision, and you would look at their brain after they died and see if if we could find patterns in brain areas. So it was slow, correlational, couldn't be experimental, and it was hard to learn things over a long period of time, but this was the observational tool that we had at the time. Later on, we could create brain lesions in lab animals. We could damage specific parts of the brain, see how the lab rat, for example, acted and behaved differently, and see if that would be the same in human observations that we made. Later on, we realized that because the brain was using electrochemical signals, we could use EEGs. We could look at the electrical activity. We could even be very precise with a big cap of different sensors to try and pinpoint where the electrical activity would happen. This is also a way to measure single neurons. So like that example of the monkeys with probes in their brain, we can do that. We do that to humans. It's not very often. Usually we use the less invasive, just cap looking at skin level for the electrical activity inside the brain. We can do a lot of brain imaging now too that we couldn't do before. So CT or CAT scans are basically a 3D X-ray, and then PETs look at glucose use by cells, which tells you like which cells are using more energy and tells you what's being active at that time. MRIs use magnets and radio waves to look at the interior of the brain, as well as seeing with fMRIs what cells are using more blood and oxygen. That tells you which areas are more active. And so all of these together can give us a very good picture of the pieces of the brain, which areas are being used for different functions, where a particular person's certain network is. And that's very important. And what we've discovered from that is that humans across the world have some similar brain areas and brain structures. That while there can be differences, right, this is on average, while there can be differences, for most humans, these areas of the brain do the same functions, are in about the same area. So there are some neurochemical processes built into our genetics to move the neurons to specific areas and take on specific functions. So we will very briefly talk about each of these areas. Again, if you go into the neurobiology class, there will be much more detail on each of these areas, but I want you to get some basic ideas as we talk about them throughout the class. There's a couple key areas that come up many times throughout the chapters. So the hindbrain is the oldest part of the brain evolutionary wise. It does basic functions like heartbeat, breathing. You don't have to think about making your heart beat. Your hindbrain does this all by itself. There's also the cerebellum, this really wrinkly portion back here that does a lot with balance, coordinated movements. That talks with other areas of the brain that do voluntary movements as well. But that's pretty close to your ears where the inputs are for your balance. It's right back here, and it deals with keeping you balanced. And other things like basic alertness, survival, and that is the hindbrain. Right on top of that, so like right in the very middle of your brain, you've got the midbrain. So middle of your brain, midbrain, and it has lots of relays, lots of connections between your organs, your sensory inputs, as well as sending information to other higher brain areas on the outside wrinkly portions. So basically lots and lots of axons going to lots of areas of the brain here in the midbrain. The forebrain is basically the most recently evolved portion, and it's kind of built of two components. There's the interior component and the outside wrinkly portion of the forebrain. So the interior component houses things like the limbic system, and that has the amygdala and hippocampus. So here we see the outside wrinkly portion. Just inside that, kind of sitting on top of the midbrain, we see the pineal gland, we see the amygdala, we see the hypothalamus, the hippocampus is back here. And so all of those organs are pretty small little specific things that evolved throughout time and have some specific functions. The amygdala does things like survival, food, mating rituals, social rituals, emotions. So a lot of emotions here, some pleasure center, running some processes on things that we do. So there's a lot that's involved there. I'm going to add on here hippocampus as well. And hippocampus directs memory, recall, and storage. The memories aren't really stored here. It's kind of like the directory for where is a memory stored, or this is where the memory has been stored. It also helps with the creation of those memories, which is pretty neat. So if you were to think of the characters from Inside Out, they kind of live in the limbic system, right? They are the emotions that are running through all the processes that the character in the show, Riley, is getting. So they're there in the amygdala, they're controlling the hippocampus. So you can think of those Inside Out characters as in the amygdala and hippocampus. So memories themselves, and we'll get into this on our chapter on memory, are basically stored all over the brain. So your neurons have dormant and awake cycles, like it's not fully asleep, but less active. A better way to say that is more active and less active periods. During a event, let's say your first kiss, there's a variety of neurons active, right? Maybe there's music playing. Let's say you're at a friend's party at their house or something. This is probably not a personal experience, right? So let's say you're at a friend's house during a party and there's music playing, you're there around people, there's conversations, and there's the person that you want to kiss. So there's a lot of things going on, right? You're getting lots of sensory input. Maybe there's smells, maybe they're good smells, maybe they're bad smells. Hopefully you get some minty smells during that first kiss. But then during the event, you have sensory inputs on your lips, right? You probably have emotions happening as well as your maybe moving your arms, right, and hands. So all of those neurons, all of those networks, which we'll talk about where they are in the brain, they're all active at the same time during this event. Those neurons that are the most active during that time essentially become the memory. So when your hippocampus says, hey, I want to recall, those Inside Out characters are like, give me the memory of first kiss. Essentially, what it's actually doing is activating that network of neurons that were active during that first kiss. In a way, you are kind of rerunning the first kiss program or the first kiss memory network. You're kind of reliving that in your through your memory, which is kind of neat. But yeah, that is directed by the hippocampus. It's hard for me to keep hippocampus and hypothalamus straight because they're similar words and they're both really close to each other in the brain. So you see here is the hippocampus, here's the hypothalamus. Very similar names, very close to each other in the brain. But I'm trying to remember that the hippocampus is for memory because elephants actually have really bad memories, but hippos have good memories. So the hippocampus is the memory, direction, and recall for storage, all that kind of stuff is because hippos have good memories and elephants don't. So the hippocampus is about memory. So amygdala is a little easier to remember because it's not got as many similar structures in the brain to think about. The thalamus, again, is very near there. It's a lot of the stuff in the very middle of the brain are relay areas. Lots of signals coming in, lots of signals going out. And so that is what the thalamus is doing. A lot of relays and integration of information. Basal ganglia, a lot of coordination of voluntary movements. So you need your balance, you need the basal ganglia, you need the motor cortexes that we've kind of talked about with the macaque monkeys, but also we'll talk about in a minute. So hypothalamus, the one I always confuse for hippocampus. The hypothalamus is a pleasure center of the brain. It's not the only pleasure center of the brain, but it monitors like the body's homeostasis. Is it excited? Is it not excited? Is it stressed? Is it not stressed? It's very connected to the pituitary gland to both read and deliver hormones to our endocrine system. And it's built on rewards as well. It looks for like rewards from eating, drinking, sex, emotion. So that is what the hypothalamus is doing. The cerebral cortex is basically the outside wrinkly part of the brain. It is the newest in terms of evolutionary time. So there are some other mammals that also have cerebral cortexes versus if you look like at a snake's brain, it's mostly all hindbrain. So this is the outside wrinkly portion. We've got a bunch of different lobes that have basically their own certain function. The occipital lobes are for vision here at the back. The temporal lobes, hearing, memory. So if you remember someone saying something, that memory is partially stored there. You've also got the frontal lobes, where essentially we have our personality, our intelligence, our our active agentic choice to move and do things. The prefrontal cortex is at the very front of that frontal lobe, and it does the planning, the reasoning, the self-control. So you might have a habit. If you want to break that habit, you're going to be using the prefrontal cortex to say, don't do that thing, that kind of thing. This is Phineas Gage. He worked on a railroad. This is a railroad tie. He was putting these into the ground, and there was dynamite involved, and an accident happened, and the rod went through his chin and up through his frontal lobes. So based on that, we had some data of what the frontal lobe does. And people described his personality changed dramatically because of this accident. He did survive. This is him afterwards, after his recovery, holding the piece of metal that went through his brain. He did survive this incident, but he was very different. Before, he was a reliable worker. People said he was a nice guy. After the accident, he was basically a jerk. He was shallow, distractible, wouldn't follow directions, often wouldn't come to work, all sorts of things like that. So it really went to show us that this frontal lobe was a big part of our personality and where that is kind of housed within our brain. Then again, prefrontal cortex, the really specific higher order functions. So this really separates us from some of the other chimps, is our prefrontal cortex is much bigger and we can do a lot more planning, reasoning, self-control, metacognition, thinking about thinking. Even apes that we've taught to use language don't seem to think about thinking, or at least they don't express it in their sign language. So with that, in this cerebral cortex, the outside wrinkly portion, we also have the somatosensory cortex. This is when we get sensory inputs from around the body. And this really funny picture here really demonstrates how much brain area is dedicated to each part. We've got a lot of brain area for feeling things with our hands. We've got a lot of brain area for feeling things with our mouth and our lips and our cheeks. We also have comparative size-wise, a lot more area devoted to our genitals in the brain. You can see that here as opposed to, say, the legs. The legs are a much bigger area of the body, but there's a lot of brain area devoted to like the genitals, the face, and the hands, and specifically fingers as well, to get sensory information from around us. This also might explain to you why some babies like to put things in their mouth. They've got a lot of sensitive sensory inputs there in their mouth to learn what this thing is, right? So giving them toys that they can safely put in their mouth is a very good plan. So big blocks rather than like little things, because it does have a lot of sensory input and their brains are trying to learn new things. So that's the sensory cortex, and then right next to it is our motor cortex. Similarly, this is areas of the brain that are devoted to specific parts of the body. So again, we've got a lot for the face, because that's very important for like eating, drinking, swallowing, that kind of thing, and a lot devoted to the hand. So if you had something like, say, the loss of a hand, you might be able to have some substitution of function and have maybe these areas for, like, you might have seen people who lost the ability to use their hands, but they learned to, say, play piano with their feet. Well, maybe that's substituting that function over here. So brain plasticity. Another important part of the brain is near that midsection is the corpus callosum. It's basically the connection points between the two sides of the brain. Tons of axons just going back and forth, sending messages across, because anytime we're doing things with our brains, we are using a large part of networks in our brains. Lots of neurons going all the time, and a lot of that information travels through the corpus callosum and through some of these other organs that we already talked about over here in the brain. So with this, there are also the spinal cord is part of our nervous system, right? It's sending messages and receiving messages down through our limbs. We've got the somatic nervous system that's getting sensory information from, say, our hands or our feet. The autonomic nervous system that's really related to our organs from our spinal cord, helps us calm down, helps the body focus on, or that area is focused on like keeping the body at homeostasis. And then as well as that, we have I'm going to jump forward a little bit here. We've got our endocrine system. So this one is often forgot about when we talk about the body's communication networks, but the endocrine system is an integral part of our body and our brain communicating with itself. So the main areas that the brain connects to it are here in the hypothalamus and the pituitary gland and the pineal gland. So the pituitary gland and pineal gland can release hormones. The hypothalamus kind of directs the release as well as reads the hormone levels in the blood. So this is a little different than the neurons because essentially you're pumping chemicals into the blood. Once the blood fills up enough with the chemicals, the organ like the pancreas or the ovaries or the thyroid can receive that message and do something with it. So the endocrine system is that system of glands. It regulates our body. It sends messages to our brain. Our brain sends messages to it. And the glands are basically just the organs that create the chemicals that control body functions. So a couple of the different ones really quickly. And hormones in general regulate behaviors such as aggression. It's involved with mating, parenting. It's involved with behaviors, and your hormones can be influenced by your behaviors, and you can influence your hormones. So you've ever been stressed and calmed yourself down, or had something, or gotten yourself more stressed out, your brain is affecting your hormones are affecting your brains. And so the study of psychology in the endocrine system is called behavioral endocrinology. And with this, the main point of the endocrine system is that it's slow. It's a lot slower than neurons. Neurons can fire 200 times a second. For the endocrine system, we're kind of down to what the body can read in the bloodstream. So for the pancreas, for example, it does a lot of glucose and insulin production as you are eating. So you're eating, you're filling up your stomach, it starts to release some glucose and insulin. Once you're full, it starts releasing a lot, but you need to get to a certain level in your body for it to be read by your brain. So that hypothalamus detects it about 15 minutes after you're full. So if you've ever heard someone suggest that you eat slowly, that's basically why. Some of these hormone systems are that slow. Some, like the fight and flight response, can be very, very quick. So you've probably experienced that. Someone jump scared you. You get really stressed out. Your muscles get tense. You've got a lot of adrenaline, and your body's charged up with those hormones to help you act. Or sometimes it makes you freeze, right? If you're charging up your muscles and you're not sure what to do, or even if you want to do something, your hormones might be telling your muscles to do something but not do something, and then you're kind of frozen there. So it's a pretty common thing as well. So the endocrine system is part of the neural system, is part of the nervous system of your body. So some of the glands, pituitary gland, it controls growth hormone, regulates a bunch of other glands. It's kind of like with the hypothalamus, a boss of glands. The adrenal gland, it regulates mood, energy level, helps us be ready for action or alerts other neurons and glands. So it secretes norepinephrine, which, as we remember, is a neurotransmitter. The pancreas does a lot with digestion, hormones like insulin. It's relevant to the metabolism, the intestines. So the ovaries and the scrotum are both useful for sexual development and reproduction, and it also is involved in the production of secondary sexual characteristics, like during puberty. So the growth of breasts or the growth of beard hair. So these kinds of things. So that endocrine system is connected to the brain. It is all part of the body's communication system. The hypothalamus, pituitary gland, pineal gland, they're getting and detecting, and a lot of organs can actually detect hormones and get like orders from the hormone levels. So it's all a big communication system of how your body works together to do the things you need to do, whether it's needing to go and make yourself dinner, or whether it's that you need to go to sleep, or that you want to have a kiss with someone, right? There's a bunch of things that your brain and your hormones are doing together all of the time. So it's all a big system working together. Hopefully you have a lot better understanding of how the hormones communicate in your body and how your neurons communicate. We focused a lot on that today. So make sure as you finish this lecture recording, do double check your syllabus to make sure what chapter we are going to next so that you make sure you are reading the correct chapter. Depending on the class that you are in, we may be skipping a chapter, we may be going to the next chapter in the textbook, so just double check your syllabus to make sure which chapter you should be on.